online to a CCB seminar here on clinical record analysis. It's my great pleasure to introduce the speaker for today, which is Sean Murphy, who is currently the Director of Research Computing and Informatics at Partners Healthcare, and also the Associate Director for the Laboratory of Computer Science at the Massachusetts General Hospital. And Sean will talk about today of leveraging the healthcare enterprise for research and discovery. So without further ado, over to you, Sean, and we're looking very much forward to your talk.
Thank you very much. Thank you. And great to see everybody here today. The story which I'm going to tell today is how kind of we invented something called informatics for integrating biology and the bedside. It was been a 20-year effort occurring over multiple stages with hundreds of contributors. The original proposal came from some work that we did on the MGB Enterprise Research Repository, then known as the Partners Research Repository, which is a very large number of patients with many billions of facts associated with them in a large research database. And over time, more and more different kinds of data came in. In fact, the first data came in with Octo Barnett and his invention of CoStar. And then as we kind of went through various stages of what was called Mini-AM at the Brigham, there was the longitudinal medical record at what was then Partners Healthcare, and now Epic at the Mass General Brigham. Now, this all was put into something we called the Research Patient Data Registry, which was actually the brainchild of a very, very famous cardiologist, Gene Brunwald. And he was looking for ways to accelerate clinical trials. And data was just becoming something that could be used to find patients and patient cohorts. And so we created this repository with an interface. And the interface had a built-in query tool. And the query tool allowed folks to query the repository in a HIPAA-compliant way. And we'll go into that a little bit more in just a minute. But it allowed all that data, inpatient and outpatient, all the different patients and their diagnoses, medications, genomics, procedures, labs, to be queried to find a cohort with fairly complex queries. And then I think my plug is just coming in and out or something. With fairly complex queries. And then, but the key thing is, it only shows the number of patients in the cohort when you do the query, and it's not even the real number, it's the obfuscated number. We blur it. Small amount, usually plus or minus 3. And so you'll see on a lot of the queries, plus or minus 3. What that means is that the queries are being obfuscated so that you can't drill in and use the criteria to find one patient. Now, the query tools evolve, and we'll talk about this over time, but very importantly, these kinds of query tools against a repository, giving you an interface to use it, as a non-expert, but also to the ends of making sure that you can't go in and find single patients and violate their privacy. Once you have found a cohort and you can do a study, the idea was then you and get detailed data, but you have to do it under a specific IRB protocol. So you must go in, you must write a very extensive protocol often saying that you want access to just that cohort that you found using the query tool, maybe 500 patients, maybe 10,000 patients, and their specific data, and that you know how to take care of data, and that you understand the rules, and you have to take tests to show that, and pass those tests so that, people know that, you're qualified and know the law. And then we will give out specific data on that cohort of patients so you can do studies, and very extensive studies, genomic studies and so forth, including diagnoses, reports of all different kinds, and imaging as well. Now, we talked about we were going to go into what is the security and patient confidentiality that was behind the RPDR. Well, so patients at MGB are added because the HIPAA law says if we give them a notification, right, that states that their data will be used for research. Now, that's not going to get through all the hurdles. That's just going to go through the HIPAA law. Then we anonymize the data at the query tool so that aggregate numbers are obfuscated and prevent identification of individuals. We always are able to review all the queries that are being done at the query tool level so that we can know, in fact, if people try to break the obfuscation by doing a repeated query over and over, it'll actually lock them out automatically. And we will actually have to go in and unlock them once we're convinced that there was no malfeasance. We removed data that typically belong to the enterprise. And all of the folks that are querying are at the top level established medical investigators, so that they have commitment to research, and they can have a team of folks that work in their work group, but they're all led by established medical investigators. As we discussed, only studies specifically approved by the Institutional Review Board, the IRB, are allowed to receive identified data. And that's the second step you need. So one, you need to have given people the HIPAA notification. And second, if you want to give identified data, then you must go through an IRB. And the IRB basically is the agent of the US government to uphold the common rule. The common rule, an important set of rules that were put into place in the 70s in order to make sure that human subjects were always protected when they engaged in research. And that's what requires this IRB application to be filed to see the identified data for research. And then finally, there are special controls that we put in regarding certain kinds of data that are state controlled, like HIV antigen tests, data from substance abuse rehab programs, and so forth. So a very extensive screen that we go through in terms of how we use the data and how it makes it available for research. Yes?
Nathan has a question.
Okay, what's Nathan have to ask?
Hi, Sean. It's Nathan Palmer. I'm sorry I couldn't be there today, but I came down with a virus over the weekend.
Speak up a bit, Nathan.
My question is, what is ACE data in your fourth bullet point here?
I'm sorry, say that again.
What is the ACE data that you refer to in the fourth bullet point?
What is ACE data? So ACE data is data that's from the covered entity. You might have lots of data that comes in and you put into a database. But if you want to use your HIPAA, the HIPAA notification, it's really given under the presence of an entity. An entity is a legal construct, and it is a group of hospitals or health centers that are all under a legal umbrella, and essentially guarantees that they receive a common HIPAA notification. So what it really kind of ties back to is that if you're in the legal entity, you receive a HIPAA notification and you're guaranteed to have received it. If you're not, even though the data might be in your database, you don't know what folks received. And so that's why we only use the data that's from the legal entity. And that includes about 17 different hospitals in the Boston area, as well as a lot of different community health centers. Thanks, Nathan. All right, now with all this data, part of the question becomes how do we actually get it all into a database that can be queried quickly? And the theme that I'm going to really talk a lot about is multimodal data. There's data from all different types and places that has to go into this data set. So it's not just going to be electronic medical record data. It started as electronic medical record data, but quickly for research, the trick was that we wanted to combine the data together, right? From all the different areas of research that these patients or participants in a clinical study might have. And so to do that, we really adopted the STAR schema for two reasons. Number one is it's a very moldable framework. And that is, the key thing is that you take every piece of data about a patient or a participant and you make it into a fact. That means, so if I have the observation at one point that a patient was given a medication on a certain day, then the fact will be that kind of those things put together into one row. Patient given a medication on this date and time by this provider. Diagnosis on this patient of this type, diabetes, let's say, given on this date by this provider. Laboratory on this patient of this type, let's say it's a serum sodium on this date, ordered by this provider. And then that has a value associated with it. So then there's a set of values that are part of can be a part of each fact as well. If you really think about it, just having a diagnosis in the fact table also has a value associated with it, true or false, right? Usually you don't express explicitly the true part, but that's kind of inherent in the fact. So everything, this patient has a variant, this patient has a imaging study. All of this is digested into facts. And these facts make up a central table. Now, some of the advantages of putting all of your facts one by one as the atomic entities into a large table is, one, your table looks pretty simple, right? So it's not too difficult to kind of go in and do your analyses. Number two, you can put an index on it. So an index is the key to running facts. And if you have many, many different tables, each one of them is going to have its own index, and you're going to have to join the different fields across the tables whenever you do a query. Now, the problem is that oftentimes, if you have, like Epic, 10,000 or 20,000 tables, and you want to do a query against that data, it's going to take a very long time doing the computation, because the index lookups are going to take a while for each table. Now here, the index lookup is going to take several milliseconds, but there's only one table, so it's going to search the entire database using that one index. And it does that using these kind of binary tree searches, right? Which is kind of how you organize all your data in, let's say, alphabetical order. And then you look up, the word dog, right? And what do you see? Well, let's see, you go to the middle of the dictionary and you're like, oh, I got to the K's, I'm too far ahead. Then you go back like maybe halfway, you're in the C's now. So then you go up a little bit. Oh, well, I'm in the DRs, and so I have to go back a little bit, right? But it actually doesn't take you too many steps before you actually get to dog, as opposed to going to every single page, right? Which is what some databases actually end up doing if they need to do a scan. So if you had to do that with 10,000 books, you can see how that would take a lot longer than doing it with one very large book, right? Doing it with 10,000 little books is much longer than doing it with one very, very long with all the same data. That's the idea behind putting all of the data into this star schema, into very large table as atomic facts. And so lookups in the database can be very, very fast. And you can and do queries against your 5 billion facts in your fact table in between 500 microseconds and maybe and 5 milliseconds. So very fast. Yes.
So, sorry, just to make sure that I understood. So you're basically generating indices for these facts. You're generating these indices for these facts based on, you'll do the pace, a date index, or you'll do a provider index, right? Because if your basic thing that you're storing in your database are these sentences, right, that have like different, I would say, more primary atomic concepts, right? Like a patient or a provider or a date. Are you also?
That's right.
Okay.
So, so the question is, what do we put our index on? What are we looking up our people with? So you can use any of the columns or combinations of columns to create an index. However, the most popular index is the concept that's in the fact, right? The fact that they have diabetes, the fact that they got their serum sodium, the fact that they're put on metformin, right? Because oftentimes they're looking for all the patients who have diabetes and were put on metformin.
Right. Okay.
And so that's the most popular index.
And can you comment on how different this approach is from other large registries? Like, is this like the common approach that people who construct registries take, or is this something that is unique to what you folks are doing?
Right. Great question. So is this a unique approach? And at the time, which was at the late 90s, this was a unique approach. Now, since then, most very large databases, and that I would say, I2B2, OMOP, and PCORNet all use this concept of fact tables. OMOP divides it into like 7 or 8 fact tables. PCORNet divides it into 10 fact tables or so by, they like to keep all their diagnoses in one fact table and medications in another fact table, as opposed to what became with I2B2, we put it all in one big fact table. But there's not 10,000 tables. Now, other approaches to doing queries are, so there's a hierarchical system, which was CoStar. Epic is built on a hierarchical database. If you search by patient, it's very, very, very fast. And you can update it very, very, very fast. It's very, very good in terms of being updatable, because you can go right in and update the tree without modifying a whole bunch of indexes. However, if you want to look up data, it's very, very slow if it's something other than by the patient. Then there's something called a graph database. So a graph database is, think of it as kind of similar to the internet, right? Where you kind of traverse from one node to another. Graph database is good. It's very flexible. If you have a domain, you can sometimes put things in that domain and make an index on it. But in general, because of the way it's kind of strings together, it has to follow these paths. And so it's relatively slow. RDF databases, for example, back in the late 2000s, early 2010s, were kind of looking at doing this, but they're just currently they're very slow. Now, there's really interesting new concepts on what kind of fact tables to make, and something we've been looking at and probably will adopt at some point is a vector database as your fact table. Just it gives you more longitude in how the meanings of what you're looking at. We'll look at that a little bit in a minute. But in general, for really large databases that you want to do analysis on, that is, you want to read them, this is the approach that most folks use. If you want to write to a database, you want to update the database, create a transaction system, not an analytic system, but a transaction system, it's actually better to have many tiny tables because it costs you a lot to update the index. And so every time you update the index with something like this, we have to actually, so we update with about between 500 and 700,000 new facts every night. To do it, we actually shut the database down around 2 a.m., remove the indexes, update it with the data, then bring it back up around 4:30 or so and re-index everything. Because if you did one at a time, 100,000, a long time to update those indexes 700,000 times. As opposed to if you had things in 10,000 tables, and all you have to do is update those little tiny indexes on those tables, it's almost instantaneous. And so that's why transaction systems work well for what they do, which is, run hospital operations like Epic, and analytic systems do good for what they do, which is analyze large code.
Yeah, makes sense. Thanks.
Okay, so this concept of an RPDR evolved into this informatics for integrating biology in the bedside, because the NIH was very interested in how do we create new databases that can hold all these different kinds of data, number one, and number two, be really. So we adopted this by building software for explicitly organizing and transforming the person-oriented clinical data, like we did with the RPDR. And the design of I2B2 is very, very similar to the RPDR. It uses a star schema at its core. But it also adds this portable and extensible application framework. So it actually has a modular pattern that we're going to look a little bit at here and there that allows you to have additions without disturbing its core parts. And all this is available as open source at www.i2b2.org. The community software is all distributed as open source. You can go to community at the very end, and you'll be able to see all the different projects and all the different cells that have been built. That's what we call a module in I2B2, a cell. Because the whole gamut was called a hive, like a beehive, and so every module was called a cell. And we have different cells that allow different things to operate. And we have many different kinds of plugins and so forth that work as cells. Now, what do these new cells do? Well, they do things like enable natural language processing. So here you can see we have a lot of the informatics community that are really focusing on doing extracting knowledge from notes. Now, there's actually natural language processing, a big field. And so it has, New York Times kind of gets analyzed by all these kinds of processing units and so forth. And you'd be like, well, let's just use those. Well, the problem is that a medical note is much different looking than a New York Times article. And it uses things like funny language, uses tobacco. Who says uses tobacco except a doctor when they write a note, right? Non-smoker, negative for tobacco. And then we have this weird 50-year tobacco pack history thing. And then you find weird things like unclear smoking. What's that? Well, that's when the resident didn't ask the patient, but it's too late to go back and ask the patient. They have to put something in there so the attending doesn't. You've got social reasons too, as well as the usual kind of, tobacco embedded in words and so forth. So it's its own thing. And so modules are built in order to do these natural language processing, which is a very important part of extracting information. And in fact, these days, we have a major project using large language models. And what the large language models are looking to do is actually use the meanings of the word. So a new thing in natural language processing is to have something called word embeddings. Word embeddings use an age-old concept, right? That you have the word cat, but cat has lots of different aspects to it, right? They're furry, they're alive, they're about as big as a bread box, it's got all these different like dimensions to it. And if you're going to look up like, I want to look at small animals, well, the word cat's not going to help you at all. But the word cat with all of its word embeddings, which is a vector, sometimes 512 long, sometimes 10,000 long, as is ChatGPT, they will capture all these meanings. And so if you put all your words in all of your medical language and all your notes into a vector database, you can look it up by its meanings rather than by the actual words. And that gives you a lot of power to find new stuff. And then once you find the documents, you can actually throw them at large language models per se to give back answer a question. And so here, for example, is where a question was actually asked of a medical record using the Flan T5 large language model. This is all done on-prem. So none of this uses ChatGPT or OpenAI, right? We actually build large language models and have them on premises. So all of this is kept on in-house computer. And here the question is, I'm going to give you a prompt which includes a discharge summary, and I want you to tell postpartum hemorrhage. The questions are, what's associated? What seems to be associated with postpartum hemorrhage? And it actually starts to give answers, right? And these answers that it gives can become a fact. So it's a new way, right? So in natural language processing, non-smoker would become a fact about the patient. Now the answers to large language models can also become a fact. Right? But still, if you want to do this at the huge scales that we're doing it, you have to make everything into a fact. And that's we call the integrate this whole data integration. And we're going to integrate notes like we saw. Of course, just the structured data that comes out of the billing. We're going to integrate genetic. I'm also going to show you where we integrate survey data from the patients into the same framework, as well as biobank data, including all the consent and the genomics and the variants, plus some derived data, like that we get from natural language processing, and that we can then make into what we call validated phenotypes, the ultimate, right, expression of a fact about a patient is something that's computed, and we actually know the error bars around that computation. So let's talk about cohorts for a little bit. So there's a RECOVER study that's currently being done. It's to understand what is leading to post-COVID disorders. And to do, there's a cohort of 40,000 participants. And those 40,000 participants get surveys, extensive surveys on what it is that they're suffering from, how's their day. Every 3 months is a survey. Then we have mobile health data, which also will go in and collect data. Then we get their EHR data through FHIR interfaces. Then we get their imaging data by going into the hospitals. We get all this data, but we need to put it together into a common place. And so, for example, with the participant EHR data and the participant sensor data, we actually get this all put into a I2B2 stack. And that enables us to be able to use all kinds of interesting analytic tools to go in and analyze the data regardless of where it came from. All the REDCap data, which looks like just these surveys, right? Have you had COVID? Yes, I suspect I had, but well, that nuance is important. That's a different fact than, yes, diagnosed based on a positive PCR test, right? Still a valid fact, but it's a different fact, right? And so all that then can go as well and go through into the I2B2 stack and be used for analysis. So this data harmonization is an important property that I2B2 has. It also enables these high quality phenotypes to be generated for things such as genomics, right? So over on the right, you'll see you have a query tool. This is the first time I've kind of been exposing you to this. So I'm going to go through it just a little bit. So on panel, you have all the different things that you can query by. It includes all the different types of facts. So you can query by all people with genomic data in this case, and it even tells you how many there are just to give you a sense, right? 9,000. Here's where it has the demographics. So you can query by consent information. Did they consent? What did they consent for? Did they consent to be recontacted? Here's some of what we call curated disease, computed phenotypes. People with asthma, people with bipolar disorder, people with breast cancer. Then we can even tell you what's their positive PPV, right? How accurate did we do the computation? Because you might want more specificity and less sensitivity, or you might want more sensitivity and less specificity, right? So we give you those options. All of these are just different ways of being able to do this query. So you drag something like from the healthcare data, primary dilated cardiomyopathy, and then from the curated phenotypes, congestive heart failure. The reason we have this as a curated phenotype is congestive heart failure typically is a terrible billing diagnosis, less than 50% accurate. Billing diagnoses tend to be really inaccurate unless you really massage them using these types of algorithms, and I'll show you. But then you can also pull in something like gene, and here contains the Titin homozygous variant, right? And Titin is a protein in the heart. And so if there's a defect in that, you would expect perhaps they might get congestive heart failure. And so this query is trying to find people who have dilated cardiomyopathy, heart failure, and they have this problem with their gene. It says 70. Notice it doesn't say 70 plus or minus 3. It just says 70. This is actually a real number. The reason we can give back real numbers in the biobank is that all the patients are consented. This is different than the RPDR, where the patients are not all individually consented. They've had a HIPAA notification that their data is going to be used for research, but they have not consented. That's why we can only show those obfuscated numbers for those patients until somebody goes to the IRB, the Institutional Review Board, and gets permission, basically from the common rule of HHS, to see the identified data on a small number of patients. Here, we've already complied with the rule by getting the consent of the patients. And the consent of the patients explicitly says that we can show the limited data sets, which we have. And that's where the 70 is, not plus or minus 3. Yes?
Biobank, then the next step will be to kind of go to the data access committee or something and say what specifically you would want to do with that cohort and request full access to it. So it's not that you'll have to go through a full IRB protocol, but now that you know that these patients exist, and if you want to do more deeper studies on their patient level de-identified data, what happens next from
Great question. So what happens now here, since we got some more specific numbers, what do we do? So if we want to go and get their limited data set, which is a de-identified data set, essentially, that still includes two things that usually you can't put in a de-identified data set, which is dates, actual dates, not just years, but actual dates, and zip codes. All right. We can actually release this to MGB investigators,
right,
without going through any new committee. Now, if we want to give the names, that's different, and we have to go to the IRB. Yeah.
okay.
Yeah, versus all that kind of, that's the IRB.
But the limited data set, that's like automatically, once they're an established investigator, they kind of go through their regular trainings and attestations and so on.
That's right.
Okay.
That is correct. Yeah. So how do we compute these phenotypes? In this case, I'm going to show you how we compute some diagnosis. So what we found is, like you might imagine, that most diagnoses occur by mistake in many patients. So it turns out if you query for diabetes, that you will find the patients who have diabetes as a billing code do not have diabetes 50% of the time. 50%. Now, you might be like, well, what? That's crazy. Why would that be? Well, it's because even to be billed, even to get paid for a glucose tolerance test, which is a test for diabetes, you have to give a patient the diagnosis of diabetes. Otherwise, you won't get paid. All right. So, and then you're like, well, that's just diabetes. There's other things. What about rheumatoid arthritis? Well, a lot of folks will actually, by mistake, occasionally give people osteoarthritis, because so much who have osteoarthritis, the diagnosis of rheumatoid arthritis, just once or twice, but it happens, right? So what we find is that these diagnoses and kind of a little bit randomly scattered amongst patients. When you have millions and millions of patients, the chance that they're going to get that is actually pretty high, right? By mistake or for some weird reason. However, patients who really have the diagnosis tend to get more of them, right? Every visit, they'll get that diagnosis. Every visit, it'll say diabetes, visit, diabetes, visit code for diabetes. So every single visit, they might have 30 visits, they'll get a code for diabetes. So you could say, well, just split it and say anybody who has less than 2, or 2 or less codes for diabetes, just say they don't have diabetes. And if they have more than 2, then they do. Well, that's nice. But what if a person just came into the hospital, right? Or has just been in the hospital for a few visits. They have diabetes, but they've only been here for like a couple of months, right? All right. So clearly you have to correct it. Just same idea, but just correct it for the number of visits they had. That's the whole idea of phenorm. So if we split, here's our patients, the blue is our patients who got the diagnosis by accident. See, and then we get a little bit, right? The red is the patients who really have the diagnosis, and the ones over here are the ones who've only been in this hospital a little bit, right? So that's why they only had it a few times. Now correct it for how many visits they've had, and you can separation in the 2 curves. And here's the key. You can actually, using math theory, separate those 2 curves without an intervening doc, right? Saying this patient has this and this patient has that. And that lets us do this in an automated fashion, and we can generate hundreds of these computed phenotypes. Now, we do go and test them using gold standards with docs, right, to make sure that we know what the error bars are around them. But that really brings us into this realm of, with I2B2, we can present these very accurate diagnoses. And so we present these all in charts. And so you can see things like eating disorder, right? Very poorly captured in codes, well captured phenotypes. All right. MGB Biobank has been a major use of this data. And I'm going to just show you kind of what the Biobank portal looks like. Again, all of the survey information that we collect with the Biobank, confirmed health information, and even special new information that we were collecting during COVID could all be a fact and all go into this left-hand side. Over here, this is a Venn diagram. Each square is trying to be a circle, essentially. So if you put something in the same circle, let's say hypertension and diabetes, you're going to get all the patients who have hypertension or have diabetes. Probably not what you wanted, but that's what you'll get. If you want all the patients who have hypertension and also have diabetes, you'll put hypertension and diabetes in that. And then you'll say run query and get the results. And so, for example, here, we're looking for all the patients who have a confirmed case of COVID and chronic kidney disease. And then we run it, and we get 722 patients in the biobank, right, who had that. and then breakdowns. Now, remember, here we don't have to have plus, in the RPDR, there's plus or minus 3 on every single one of these, and it blurs them all. In the biobank, we don't have to do that. That's a big advantage to having consented data in I2B2. We can also do things like special kinds of causal analysis workflow. We can do the basically a timeline of their data. And then without getting the patient identifier, just this patient num, we can actually extract data and people can use it directly. Now, every time you use the biobank, or when you sign up to use the biobank, you have to sign this big data use agreement. That's part of what the consent says. But once you've signed that data use agreement, you can actually get the limited data set directly. And this concept of being able to get the data directly is really important. And we provide it when we can, because we can't do it with the RPDR. You have to go through the process of going to the IRB and then you'll get the data, right? But here in the biobank, we can provide it either as a wide file that is, your classic, column for diabetes, yes, no, column for, are they on metformin, yes, no, right? Or a tall format, just like the fact table looks like, right? One fact after another. Or as flat files. And sometimes when we have images and so forth that we have to export, that's what helps. And then you can put it into these environments, right, that we're all familiar with doing computation, like Python or R. Now, in the situation where we don't have the consent, what we've been working through is creating a limited data set, but putting everything in a data enclave. The problem is that we want to make sure that you can't take the data away, right? Now we want to make sure we can, in a situation, send it to patients, we allow the data to be taken away. That's what the data consent essentially says, is that we're going to distribute the data and it's okay for people to take your limited data set and use it for science. But in a place like Mass General Brigham, where they've consented, they have agreed, right, to the HIPAA notification. They will need, we treat their data differently, and we keep a limited data set in a data enclave, and only very specific folks can go in and use the data. But then they can get the data off I2B2 in the same way in tables and so forth without going through that process. Now, again, you can't get the identities of the patients here. You have to go through the IRB no matter what to get the full identities of the patient, which might be what you need if you're doing a clinical trial, for example. So this helps for some. What it does is it really lets you think of I2B2 as a big data index, right? In order to get to lots of different kinds of data. And that's really important. So you can't get their identities, but you know who has what patients and how many patients have what, and you can look at it inside the enclave. And then if you really need their identities, you'll go to the IRB and get them. So that's an important concept of how IRB has evolved kind of over many years, right? It started as kind of like a obfuscated data set. Then it became a data set for consented patients. But then as we wanted to put lots and lots of different kinds of data into it, we started thinking of it more of as a data index, some of which you could use in the data enclave, and some of which we have IRB approved to it. For example, if you wanted the names and addresses to use for recruitment, then you would need to go through the IRB. Now, another interesting thing that evolved from I2B2 was because it was all a distributed query system, that is, it uses these modules, we could actually put it at various hospitals and let people do queries against all the hospitals. Now, they also have this plus or minus 3, right? So you must have a obfuscated count in this case. You're only going off of the HIPAA notification. You haven't put it in an enclave, so you're going to go off the obfuscated count. But still, you can get more or less in this case, how many patients at each hospital do you have with hepatitis C if you want to do a hepatitis C study and know which hospital to go to for it. You'll do the query in what we call the Shrine Hub. It'll go to the different nodes, give you your answer in the Shrine web client. And then if, for example, you're one of the sites and have a lot of patients with hepatitis C, it looks like a great site. Then we go to the individual hospital and go through the whole process at the hospital to get to their I2B2 at the hospital. So we don't allow any limited data sets. We certainly don't allow any identified data sets be directly obtained at the network level. Everything, once it gets past this initial query, goes and is done at the hospitals, and that's where you can then get the data. Now, coming up, we've got a new I2B2 query tool that's getting released. It does the temporal queries really nicely, and it kind of lays it out really nicely. And we also have a new I2B2 AI query transform coming up. So you might be like, well, okay, but what if I could just type in the query and then it'll just, kind of distribute it into the query tool and do the query for me, right? And that is what we are currently working on that. Basically, it'll then the I2B2 query, so you can know, oh, that's right or it's not right before it goes and launches it. But the idea is it makes it easier for you to get started. And we have over 300 implementations of I2B2 across the globe now. So in summary, I2B2 is specifically good for multimodal patient data because it kind of uniformly puts it into these facts, and that way it all becomes interoperable. It can be used to index the patient data at a massive scale like RPDR does with 5 billion facts. Many varieties of harmonization can occur in I2B2 to produce data sets for analysis, and automating the flow of data to a large set of really diverse endpoints lets you really enable the learning healthcare system in this manner. And then here are just some URLs that you can go to look up more stuff about I2B2.
Well, thanks, John, for a super interesting talk. I think there were already some questions during the talk, but we have some more time for questions. Are there any questions here in the audience or online?
Yeah.
Oh, it's the mic. So I2B2 is a software that you can download and use to analyze your patient data set, large data, correct?
Right.
So how difficult would it be to integrate like 10 sets of boards if you download the software? Could you just speak about how like the data upload works and how kind of it gets indexed? Like, is that a user type thing to do, or does the software take care of a lot of the features?
Yes. Right. That really gets to how do you load data into I2B2? And overall, it is a process, and it is largely left up to the person that's creating the I2B2 data set. We have a number of tools that you can actually find at the site that helps you with some of it. But there's a conceptual, right? How do I divide this data set into facts that really you kind of need to be an informatician to kind of engage in that? Because really what you're going to be doing is taking a classic, usually they come as an analytic data set, right? Which is a large flat table where you have one row per patient and then one column for each fact. And instead, we're going to want to pivot that so that it looks like one fact for each row. And so that process, it kind of takes you thinking through the mapping of, okay, what in that flat table would it look like if it was one be, 10 or 12 or 20 different facts. And then how would I write a program, right? And we have some sample programs, but ultimately, you would want to write a program for whatever the table looked like, fit it into an I2B2 fact table.
Okay.
Yeah, I mean, it's a really good question, and that's an important, nothing comes for free in life. Now, I have to say, we are experimenting with doing some of this using large language models. We'll see. My intuition is that they're going to get them to the 90% stage, and then it's going to be hard to get to the end of the.
Okay, and you mentioned that you're using just one in-house large language model. Are you planning on comparing them with like GPT-4 or Gemini?
Are we going to compare our large language models? And so we don't actually just use one. Each group kind of likes to my group tends to use Llama 2, because then you kind of become an expert on the different aspects of it and different variations and how it changes and how you can fine tune it and so forth. But so we've done a lot of comparisons with generic data, I'll call it, off the web. So there's like notes.csv and stuff like that. You can get and then try it in different ways. And in general, you fine tune Llama 2, you tend to get for that task better. Zero shot, you tend to get better results using GPT-4.
Okay, great. Thank you.
Now, one more thing. So a lot of using models depends on the embeddings models, right? So a lot of it is how complete is your dictionary of embeddings. So they both have fairly complete dictionary of embeddings, but I think in really specialized medical applications, it becomes more and more problematic.
Other questions online for Sean? Maybe I take one. So I totally got convinced that this is an enormously useful resource for research and discovery. One thing under the notion of multimodal data is, I saw GWAS data is in there, genetic data in there. Is there any thought of expanding to maybe like other omic modalities, gene expression levels, maybe in some sort of discretized form? Is that something that is in the planning?
So great question. So advancing to different kinds of omics, right? So gene expression levels are good because they're just kind of a concept and a value next to it. Metabolomics tends to be more complex, I have to say. And so the representations, sometimes the final results fit nicely into as a fact, but a lot of the intermediate ones are often better expressed as a graph model, I have to say. So in many ways, you use the I2B2 star schema as kind of a final dimension to put all the data together. But sometimes the intermediate steps of getting into like a final kind of digested fact doesn't always work in a fact table. And often there'll be different kinds of databases that work well. Yeah.
There's one more question in line from Nathan. You mentioned that you are looking into vector databases in the I2B2 ecosystem. Folks online are curious about what role you see these databases playing and whether you have any experience with prototyping or running any of them.
So vector databases. So the role that I see in them is that it will help you do text queries better. So for example, if you wanted to look up the people who are getting thoracic operations, right? You might do a query of thoracic, the word thoracic, right? And then operations, then put it together. Now you're going to get a lot of funny stuff, thoracic this and thoracic that. And what about people that call them chest operations, right? So you have, okay. What if you didn't put in just the words, but you put in each sentence in your OR report as all the words with all of their vector embeddings, right? So you put them in a vector database. So essentially, if you had 12 words in your sentence and you had 512 dimensions to your embedding, you would have a 12 by 512 vector database in that case. And so then you might have, few million sentences to put in. And so that would be 1,000,000 times 512, right? Now, but now you can, if you want to look up thoracic, make that into embeddings, right? So embeddings make it into the meanings, the various meanings of the word and meanings, right, of that. What it does by looking up the meanings of that is you go beyond thoracic in terms of looking, you go to chest. On that, right? Even things like, cavity and so forth will get kind of understood. To do that, you can then collect all the sentences in the medical records that have something to do with a thoracic operation, right? All you've done now is a bunch of sentences that have to do with, now use those, put those together into a prompt and send that to a large language model. And then you can ask it a question. I mean, sometimes maybe that's what you want. You want a nice, very complete thoracic operation bunch of sentences. But what we've been doing is using that just as the prompt, all those texts as a prompt to go to the next and send those to the large language model, ask it a specific question, like what sentences seem to, or I shouldn't say, yeah, does this patient seem to have had any thoracic operations, right? And then it'll look through and then it'll take into account that, there might be some no's and yeses and so forth in there about whether they have or haven't had thoracic operations and digest that into a concise answer. So that's kind of where we're going with the vector databases. It's a thing that there's a term called RAG that's kind of been coined in the lay literature for this, RAG.
Okay, excellent. Well, I think with that, we're at the hour. I'd like to thank the speaker again for a really fantastic talk and for taking the time to come in here. And other than that, I wish everyone a great day. Thank you.
Thank you all.